ELISA, enzyme linked immunosorbent assay, also known as enzyme immune assay. It is a biochemical technique to detect or quantify presence of specific antibodies, antigen or hormones presence in the sample. The sample is patient's blood serum. The amount of a compound correspond to the substrate is converted to colored product by the enzyme linked antibodies. It is a quick yet sensitive method of quantifying large number of samples. ELISA replaced radioimmunoassay because of its quick and sensitivity regarding the results. It successfully diagnosed HIV, rotavirus, syphilis, Zika virus, etc. After that, we are going to see the requirements of ELISA. So here are the requirements of ELISA. These are microtiter plate, which is having 96 wells, made up of plastic and coated with polystyrene. To that we need a compound which contain targeted antibodies, antigen or hormones. After that we need primary antibodies. This plays major role here in the test. After that we need secondary antibodies. This is called secondary antibodies because they join to primary antibodies. After that we need substrate which is going to produce color when targeted antibodies are present in the sample. Okay. After that we are going to see the types of ELISA. There are three types of ELISA, that is direct method, indirect ELISA and last is sandwich method. Okay. So here is the first method, that is direct ELISA. So what is a direct ELISA? In that we are going to see, in this method the sample is attached with well. After that primary antibodies are added and the plate is incubated. After that wash is given. When wash is given, the excess antibodies which are not bound get washed. After that substrate is added. If the target antibodies are present in the sample, the substrate produce color and from that we come to know the antibodies are present in the sample and test is positive. The next method is indirect ELISA. So here in indirect ELISA, first we add sample. After that we add primary antibodies. Then we give it incubation. After that the sample is washed to wipe out the excess antibodies. After that secondary antibodies are added and sample is again incubated. After incubation then again wash is given to wipe out the excess antibodies. Then substrate is added. Substrates get bind to the secondary antibodies and produce color. If the sample doesn't contain the antibodies, then color will not be produced. Next is sandwich ELISA. In this process, antibodies are attached with well like this. Second, we add antigen or sample and the plate is incubated. After that, wash is given and the primary antibodies are added. After that, incubation is done and again washed to remove excess of antibodies. Then secondary antibodies are added which get bound to the primary antibodies. After that, incubation is done and finally substrate is added. If the antigen is present in the sample, it produces color. Otherwise, it will not produce any type of color. So this is a sandwich ELISA. After that, there are some applications. The ELISA is used to evaluate presence of antigen or antibodies present in the sample. It is used to determine the serum antibody concentration in virus test. It is also used in food industry to determine food allergens. It is applied in the disease outbreaks, tracking the spread of disease. It is used to detect various kinds of diseases. ELISA is also used in vitro diagnosis in medical laboratories. So this is about the ELISA test.